Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Zoltan Gulyas from Thermo Fisher Scientific.
Thank you, Leah, for the introduction. Hi, everyone. I hope you're doing well, staying safe and healthy in these challenging times. I would like to talk about purification of vaccines and how these processes can be intensified with the implementation of novel, high-performing techniques such as Affinity Chromatography. Vaccination has been successfully protecting humankind for decades against a wide variety of infectious diseases. We've been able to fully or almost fully eradicate numerous life-threatening pathogens and thereby preventing millions of deaths and other serious consequences such as paralysis every year. However, the current pandemic is unfortunately the most obvious evidence that we should not sit back and be too satisfied with what we have achieved so far. Novel pathogens are constantly appearing and becoming a serious threat to human life through specific mutations, causing epidemic or even pandemic outbreaks such as the current COVID-19 crisis. It has never been more obvious that vaccine development timelines need to be shortened and processes need to be intensified significantly to have a rapid response and keep up with the global demand of millions or even billions of doses, depending on the scale of the outbreak. These goals can be met by utilizing novel types of vaccine modalities as well as introducing high-performing solutions for vaccine development and production. In order to stop the COVID-19 pandemic we are all sitting in, tremendous amount of work and resources have gone into vaccine projects and potential treatments as well. And as you can see, we have almost 200 different candidates of various modalities at various clinical stages. 5 of them have already been approved by regulatory agencies, and vaccination of people could be started after 1 year of the first occurrence of this virus, which is an unprecedented success, and no one really dared to dream about it in early 2020. I would like to take this chance to say thank you very, very much for the hard work of everyone involved in these projects. Moving on to the vaccine modalities, the landscape is relatively diverse nowadays. The first vaccines have been attenuated or inactivated viruses, and these types are still commonly used today because they induce strong immune response, and we can rely on them as we know and manufacture these modalities for decades. The introduction of recombinant DNA technology and modern biotechnology modernized the vaccine landscape as well, because the relevant subunits or the empty shells of pathogens called virus-like particles, or VLPs in short, could remove the risk of replication and sickening of people which are associated with whole virus vaccines. However, these new modalities usually require adjuvants or specific delivery systems to elicit the appropriate immune response, so there's always a trade-off. DNA and RNA vaccines and viral vectors such as the adenovirus-based vaccines are the most modern modalities, where the subunits are transferred in the forms of RNA or DNA blueprints into the human bodies. This way, the host cells are expressing the antigens similarly to the actual infection, which are then stimulating the immune system, triggering the response. 2 of the COVID-19 vaccines approved by the FDA and the European Agency use the mRNA technique for immunization, which is not only a major scientific breakthrough, but it could potentially bring a paradigm shift in the vaccination field and in terms of how we respond to outbreaks in the future. Moving on to purification, this slide depicts the purification platform for all biologics, starting with cell harvest and clarification, followed by chromatography steps for capturing, polishing, and steps dedicated to bioclearance and the final formulation at the end. Protein A for molecular antibodies has shown us the power and success of affinity chromatography in the capture step, which could be adopted by manufacturing processes of other types of molecules, including the vaccines as well. The Capture Select portfolio of Thermo Fisher Scientific offers affinity solutions for a wide variety of targets, such as antibodies and antibody derivatives, recombinant proteins, viral vectors, and even vaccines. The porous ion exchange and hydrophobic interaction resins, on the other hand, are ideal for the traditional non-affinity capture and polishing applications because they offer high capacity, high productivity, and high resolution at the same time. Why affinity? What are the benefits, you may ask? Well, if there is a process built on the traditional non-affinity capture, multiple polishing steps might be required to attain the purity. The problem with having multiple steps is not only the costs and extended development and manufacturing timelines, but the loss of product recovery as well, as you can see. Despite of high recoveries at each step, the overall yield decreases significantly with the introduction of additional purification steps. So you could clearly and very quickly lose 50% of the produced product after 5 or 6 additional steps. The high selectivity of the affinity ligands, on the other hand, can provide high purity in a single step, and only 1-2 additional polishing steps may be required to meet the quality targets. Select technology is built on camelid IGs under variable regions called VHH. These molecules contain heavy chains only, and the variable region is a single domain, as you can see, but its selectivity is similar to the standard and full IgGs of other species. The lack of light chains make these molecules more soluble and stable, and eventually enables their application as affinity ligands. We have a unique high-throughput screening-based technology where we start with hundreds of different candidates for the actual target. We screen for not only specificity, but high dilution conditions and ligand stability, as these resins are meant to be reused. The final candidate is expressed in yeast in a fully animal origin-free process, and we can scale it up to 15 cubic meters of cell culture volume if needed. The ligands are tested on both agarose and porous backbones to select the best version for the actual target. Agarose resins are usually working better for recombinant proteins and antibodies, while porous-based resins provide higher capacity for larger targets such as VLPs, viral vectors, or viruses. Most of our Capture Select resins are fully CGMP compliant, and some of them are already used for commercial manufacturing or have been used for producing clinical materials for phase 1, 2, 3 studies. I would like to highlight one specific Capture Select affinity resin called CTAG XL, which might be particularly suitable for vaccine development and production. The CTAG itself consists of 4 amino acids, and as the name suggests, it should be located onto the C-terminal end of the molecule, basically any molecule from recombinant proteins to VLPs. Given the small size of this tag, the higher order structure of the targets can be maintained, and similarly to the malaria vaccine candidate of the Jenner Institute, the tag may still be present on the final molecule, as it should not be immunogenic within the limited number of injections required for vaccination. The Capture Select CTAG XL resin is based on proper immunoaffinity interactions, which provide significantly higher purity and recovery compared to HisTag and the IMAX system, which was also demonstrated by the Jenner Institute in the reference paper. With the introduction of CTAG and CTAG XL, they could increase the recovery from 52 to 77%, and the purity from 85–90% to more than 99% at the end of the 2-step process, which has actually made the project commercially viable. The resin enables high dilution options apart from the commonly used acidic conditions, which is key for vulnerable molecules and in order to ensure high recoveries. High dilution usually means mildly acidic conditions or even neutral pH in the presence of additives such as magnesium chloride. The resin is fully scalable and CGMP compliant, and you can use it for clinical production as well as commercial manufacturing on a routine basis. We can support manufacturing with our pre-packed and ready-to-use evolved columns available in different sizes, And we also offer robocolumns because we see that more and more companies are selecting C-tag over his-tag in high-throughput screenings and rapid purification of vaccine or other molecule candidates. If you're interested in this technique, I would encourage you to review the reference publications, the article from the Jenner Institute, and the white paper on chemical and engineering. Moving on to the porous technology, which is our backbone for affinity solutions targeting large molecules such as biovectors or messenger RNA, and the basis of our ion exchange and hydrophobic interaction portfolio. The three main features of porous resins are the material itself, which is polystyrene divinyl benzene, the opened-up pore structure optimized for large molecules, and the 50 micron average bead size, which is a good compromise between resolution and process performance, especially if the resin is applied for polishing applications. The next slide shows the exact benefits of these features. The rigidity of the beads combined with large through pores enables the use of high flow rates without significant loss of capacity and resolution. As you can see on the left-hand side, resolution can be maintained even at 1000 centimeters per hour. The middle graph depicts the capacity of porous XQ and HQ resins in comparison with an agarose-based resin at different flow rates. And the porous resin can maintain capacity at high flow rates, while the agarose resin loses most of its capacity due to compression under high pressures. The pressure flow curve of porous is linear, as you can see on the right-hand side, which indicates predictable behavior and back pressure upon scale-up. This is a busy slide, but bear with me. The key message here is that we offer 2 weak and 2 strong anion exchangers, 2 strong cation exchangers, and 3 hydrophobic interaction resins with different hydrophobicities. If there is no suitable affinity solution for your molecule, you would prefer the traditional non-affinity capture, or you would require additional polishing steps, you can choose from this selection, or you can reach out to us because we are always happy to help. Our porous HS cation exchanger has been applied for the purification of human papillomavirus VLPs by Merck in the US. They took the clarified lysate loaded directly onto the porous resin, eluted the VLPs with a linear salt gradient, and they could obtain 98% purity after one step, which was a great success. They also concluded that the higher capacity was attributed to the large through pores of the porous resin. I'm sure you're all following the news about the approval of mRNA-based vaccines for COVID-19, which is a brand new but already successful vaccine modality to fight the pandemic. We've been working hard in the past years with selected customers to develop an affinity solution for mRNA molecules, and we had no idea how important this project could be at some point. The porous oligo-DT resin uses our largest pore size backbone, which is 2000 angstroms, a proprietary linker, and 25 mer of thymines as affinity ligands. The interaction is based on hydrogen bonds and annealing of adenines of poly-A tails and thymines of the ligands. The resin is fairly easy to use. You would require high conductivity to reduce the repulsion of phosphates and thereby enable the annealing of nucleotides. After the intermediate wash, you would simply need a low conductivity buffer or even water to break adenine-thymine interactions and promote elution. We have seen good capacities, namely up to 4-5 milligram per mil with smaller to medium-sized targets, up to 4,000 base pairs, and high recoveries, meaning above 90% or even 95%. The resin can be cleaned with half or even 1 molar sodium hydroxide for reuse, fully CGMP compliant, and the production process is animal origin free. And finally, just a brief snapshot of our latest affinity solution called BaccuClear that combines both Capture Select and porous technologies. I bet in Portugal we're using insect cells and the baculovirus expression system to generate influenza VLPs, but they had issues with baculovirus removal using the routine methods because both baculovirus particles and the produced VLPs had similar size and envelopes around the capsids. This challenge gave us the chance to work together with them on a novel approach where the selectivity of the affinity ligands are leveraged for scavenging purposes. It means that the impurity, which is the baculovirus, is selectively captured by the resin, while the product, which is the influenza VLP, flows through the column unretained. If you're interested in the details of resin development and process optimization, please take a look at the reference publication below. And please let us know if you're facing similar challenges, because this resin is available off the shelf and we are happy to send you samples for evaluation. I've briefly touched upon some of these formats already, but one more time. We offer loose resin robo columns and 1 ml, 5 ml pre-pack columns to support process development. We have leakage ELISA assays for CGMP compliant capture select resins to monitor the ligand leaching. And we have HPLC columns and biotin conjugates that can be used for various analytical applications and assays. And with that, I would like to thank you for your attention. Please let us know if there is anything we can help you with. Stay safe and healthy, and I'm happy to answer any questions. Thank you very much.
Great. Thanks, Zoltan. So the first question is, are you developing resin specific for the SARS-CoV-2 virus?
Yes, thank you for the question. Indeed, this looks like an obvious one to ask, and we are certainly interested in, but we would prefer to have collaboration on this project. I have not mentioned it yet, but both the Capture Select and Porous teams offer custom ligand and resin development services. And if you're interested, we are happy to work together with you on these targets, or any targets basically, including the spike proteins of SARS-CoV-2.
Okay, and you mentioned the resins are CGMP compliant. What do you actually mean with that statement?
That's a very good question, thank you. So it means the resin production process is fully validated, all aspects are validated. We developed a leakage ELISA kit for the detection of leach ligands to support quality control and demonstrate that the leach ligands are below the safe levels. And we can provide you with the full regulatory support file documentation, which is usually a multi-hundred pages long document containing all information about the respective resin.
Okay, and does the Oligo DT25 affinity resin remove double-stranded RNA molecules?
Thank you. Unfortunately not. So if those impurities have poly-A tails as well, then the Oligo DT would pick those up as well. So basically anything with a poly-A tail. And therefore, we recommend to implement a second step, a polishing step, which is typically reverse phase chromatography or hydrophobic interaction chromatography, or probably ion exchange chromatography to remove this specific impurity from your product.
And can I fuse the C tag also at the N terminus of my target protein? And does the tag impact the folding of the protein itself?
Again, very good questions. Thank you. So unfortunately, the sequence must be placed onto the C-terminal end. This is the only limitation, the bottleneck. Otherwise, the affinity resin, the C-tag XL, is not able to recognize this motif due to structural differences if placed anywhere else. Regarding the folding itself, it was probably mentioned, but it should not cause any issues because the C-tag is very small and we have not heard about any loss of functionality caused by the implementation of C-tag. So I'm positive that the C-tag should not influence the higher order structures and folding.
Okay, well, it looks like we have time for just one more question. If we didn't get to your question, please go ahead and type your question in now, or if we didn't get to your question, we will be passing all the additional questions on to Zoltan, and he can follow up with you directly. So the last question is, do you have purification resins for viral vector-based vaccines?
Yes, we do. Thank you for the question. So we have affinity solutions for adeno-associated virus and adenovirus. I appreciate that AAV is primarily used for in vivo gene therapy applications, but we have great solutions for capturing serotypes 8 and 9, and we have a solution called AAVX, which is able to bind all naturally occurring serotypes and most engineering ones as well. And for adenovirus, we have the porous capture select ADV5, which is able to bind the adenovirus 5 serotype. And finally, we have another resin called porous heparin, containing immobilized heparin, which can be used for retrovirus and lentivirus purification. But again, these are primarily used for cell therapy applications.
Thanks. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.